today, our second speaker is an expert who has this special capability. So please help me to welcome Professor Dr. Kim-Anh. Professor Likau is an academic in the Melbourne Integrated Genomics in University of Melbourne, Australia. She develops computational methods, software, and tools to interpret this big biological data and answer research questions efficiently. So Kim-Anh has a mathematical engineering background, and she graduated with a PhD in statistics from the University de Toulouse, France. She started her academic career at the University of Queensland, Australia, before moving to the University of Melbourne, where she continues her strong research focus. So Kim-Anh, the mic is yours.
Thank you, and thank you for the invitation. So the aim of this presentation today is just going to give you a bit of a flavor of what we mean by multi-omics data integration. And I will also talk a little bit about the tools that we have developed to do this task. But I just want to start by saying that I have a statistical background, but I actually work at the interface between biology and statistics. So as you know, in biology, the field is very qualitative, whereas in mathematics, it's extremely quantitative. And so really my job is to work between those two disciplines to make them talk together and also make more progress in research. And so I've been starting working in that field for more than 15 years now. And it became clear already back then that you needed a strong interaction between those different disciplines. And the reason being that we generate a lot of data now. They're coming from different sources, from different technologies. There's, of course, a lot of computational issues to foresee. What we find in statistics is that the number of samples is usually very small. And so it's very difficult to validate the results statistically. And so what we do now in computational biology is to rely a lot on the biological interpretation of the results to make sure that the analysis is actually doing well. And you've seen it. I mean, we talked about, microbiome, which is, an emerging high throughput field, but now, we're working also in single cells. So there's a lot of technologies that keep arising, and they're all doing multi-omics now. So this is my lab. We have expertise in both statistics and computational biology. It's highly multidisciplinary. So we have not only statisticians, but also bioinformaticians, data analysts, software developers. And this is how we all work together to produce what I'm going to present today. And I think a specialty or a strength of our methods is that they are often technology or omic agnostic because they are non-parametric methods. So they can apply to a wide range of different types of data. And we have funding mostly from the Australian government, but also a bit of funding from the NIH to support this work. So the reason why the field of multi-omics is quite difficult to understand or even to enter is because for many, many years, we have had this reductionist type of point of view, where basically we would have, we would make the hypothesis that, one gene is responsible for, say, a disease. And from a statistical point of view, then you run one statistical test, for example, a t-test or an ANOVA test. And when we started to generate and collate all these different types of high throughput data, then we basically are measuring thousands of molecules and variables, parameters at once. And so that requires a holistic point of view. But it also means that the hypothesis is not very well posed. It's extremely exploratory in nature, and I'm sure a lot of you who have already heard about multi-omics have heard about the fact that it is a fishing expedition. We don't really know what we're looking for, and we don't really know what we're gonna find out. So when I talk about omics, I talk about all these different technology that generate high throughput data. So for example, you can measure up to 100,000 of transcripts if you're interested in transcriptomics. You can, measure the expression and the abundance of proteins, of metabolites, of bacteria, as we discussed just before. But in there, I mean, there's so much information. I also put in the omics, clinical data, genomics data, and so on. And what we think about the biological dogma doesn't necessarily apply. And that's something we're still unsure about. We don't really know if everything's starting from the DNA really, or what is the crosstalk between, say, proteins and transcripts, for example. So there are different types of data integration that you can, methods that you can consider. They're all statistical methods. So one that is quite well known is a network-based method where basically people calculate the correlation between all these different types of variables or features that we have, and try to visualize it in the network. So I'll show you an example of that. What people often do also, and I'll show you an example of that, is this multi-step approach where they analyze each layer or each omic layer individually. For example, they do a differential expression analysis on the transcripts data, and then they identify a subset of those differentially expressed transcripts, and then they do the same for the proteomics, for the metabolomics, and then they look at the overlap, for example, at the gene level of all these different lists of omics that they have identified. You have Bayesian approaches as well, and the methods we are specializing in my lab are matrix factorization techniques. So basically what it means is really summarizing the source of information from this very big data into a smaller space. So it's going to be much smaller to analyze, it's going to be much easier. And so we do that in the broader context of multivariate analysis. The hypothesis really is that there are different molecules or features that act together to trigger the cell's response. And we really need to shift this one gene hypothesis paradigm when you enter this high throughput omic era. So I will briefly tell you that instead of looking at one biomarker at a time, we are going to look at a combination of them at once. And this is more useful if you're interested in biological pathways, for example. The data dimension reduction step is very useful to understand or to simplify a very complex system into something that we can actually get insight in. It's a very useful framework to integrate multiple sources of data. And we do also supervised analysis, which is useful if you're interested in prediction. So if you, for example, you're interested in predicting or giving a diagnosis based on the transcript data of a patient. So an example of multivariate methods you may have heard is principal component analysis, which is a very well-known tool, but I use a lot of methods based on projection to latent structures because they're useful for data integration. So this is my technical slide here, just to explain a little bit what's happening when we do data integration. So basically here, I'm representing a data matrix. So it's an Excel sheet. You have your samples in rows. And here you have all the different features that you have been measuring. So for example, here, this is a transcriptomics data set. So you have the expression of each transcript in a column, and you measure that for all your individuals. But because you've done a multi-omic experiment, and by that, I mean that you have also extracted other types of omics on the same samples, then you can also have, for example, a proteomics data set where you use mass spectrometry. You have the abundance of each of the proteins on the same samples. This is really important that you have the same individuals. And then maybe you have done a metabolomics experiment as well. And you also know that your samples or your individuals belong to different sample groups. So you know, for example, that they are in stage 1, 2, or 3 of disease. So instead of trying to analyze all these data sets together in some way, which is, it would be an enormous like it doesn't even fit in Excel. What we're trying to do is to summarize this information. So instead of saying I'm looking at 10 or 20,000 genes in that individual, I'm going to say I'm looking at a combination of the 20,000 genes for that individual. So in the end, I reduce the dimension of my data from a dimension P, which is 20,000, to a dimension 1. And so why are we doing this? So it's a bit scary because you think, well, you know, you're missing a lot of information there. We still retain the original information. We still do know what are the genes that are important really to characterize that sample. And we also know what are the genes that are not interesting. And so we can do what we call feature selection or variable selection, where basically inside the method, we are able to identify what are the genes, what are the proteins, what are the metabolites that together explain the disease status of the patient, discriminates the different sample groups, and are correlated together. So basically this is what this diagram is showing. And the way we do data integration is basically we require these dimension reduction elements to be correlated together. So that means that however I summarize this big matrix into this combination of features, it should still be very correlated with the information I have from the proteomics and from the metabolomics. So there's this constraint in the method that says, please do identify only the set of genes, the set of proteins, the set of metabolites that together explain the same biological variation in the system I'm looking at. And so, as I said before, each individual, for example, in the transcriptomics dataset is described by P parameters or P genes. And instead, we're going to reduce the dimension and describe that individual in only 2 dimensions, for example. So these are the kind of plots. And if you have worked with principal component analysis before, you have seen those kind of plots. Every symbol here represents one sample. So here, this is a breast cancer data set, and it's a multi-omics experiment. So you have mRNA, microRNA, methylation, and proteins that have been measured on those individuals. Those individuals are colored according to the subtype of breast cancer that they have. And you can see now it's in 2D. And so we start to see that, for example, in the mRNA data set, it's interesting because the basal subtype is very different from the other subtypes. And then you look at the microRNA and you say, oh, I see a similar trend to the mRNA, but it's not as clear. So the methods are designed to show you similar information between those data sets, but of course, it doesn't necessarily happen all the time because it's really data dependent. So this is why, for example, in the proteomics or in the microRNA, we see more noise than in the mRNA or methylation. So I told you before that not only we reduce the dimension of the data, but we also identify potential molecular signatures. And we can represent them in different ways. Here we use different plots to represent how those different features are correlated together. So for example, this is a circus plot. Those are the microRNA, mRNA, and proteins that we have selected through the methods. And the line here, so each on the quadrants, this is the name of each mRNA that has been identified by the method as explaining the different sample groups. And each line here is going to tell us whether there is some correlation happening between, for example, mRNA and microRNA, is the correlation positive or negative. And so you can see here, for example, there's a lot of positive correlation happening between mRNA and microRNAs, some negative correlation as well, and a lot of proteins are actually not very correlated with those features. So in the next 5 minutes, before I go that, so this is the type of methods we have been developed in my lab. They're actually implemented in a R package called mixOmics. where basically we developed a lot of multivariate methods for data integration and data exploration. And for different types of analysis. So you can see here, you can integrate 2 data sets, you can integrate multiple data sets and so on. What is good is because those methods use the same principle of data dimension reduction, the plots are very similar to each other, regardless of the method you're using. And so that helps the users actually to find their way in their analysis. And so I'm just going to spend just a few minutes giving you only one example here because we're running out of time. This is an example, and there's many examples now in the literature of researchers who have been using mixomics for their multi-omic integration. This is a paper where I was a co-author, so I understand the biology better. Basically, we call it a small big study. It's a small study because the researchers from Canada and the US were able to extract less than 1 milliliter of blood from newborns. And from this very small amount, they were able to generate a lot of omics data. So 5 different types of omics data. So those newborns have been sampled at different times, at day 0 when they're born, and then day 1, day 3, day 7. Then we have proteomics, metabolomics, and other types of omics. What is also very interesting in that study is we have a training data set where we have 30 newborns from Gambia. And we were then able to validate those results in another cohort from PNG. And so, as I said before, usually what people do is that they analyze each omic data set independently using differential expression tests, for example, differential expression analysis. So these are the results for the transcriptomics. Basically, we compared how many transcripts are differentially expressed between day 1 and day 0. So always compared to day 0. So between day 1, day 0, day 3, day 0, day 7, day 0. And then how many of those transcripts are either up or down regulated. And so you can see that with time, there's an increase of the number of transcripts that are differentially expressed compared to day 0, both up or down regulated. And it's the same for proteomics, it's the same for metabolomics. So once you've done that, then you can combine the results and say, okay, let's visualize this into a network to see what are the correlations happening between the transcript, the proteomics, the metabolomics, so that we can identify relevant biological pathways. And this is what you get. Basically, there's way too many features that are differentially expressed. It's extremely difficult to visualize anything here or even to understand the correlation structure of the data. And so this is what I'm showing here at the top. It's a single omic analysis. You calculate the Pearson correlation between the different features you have measured, and then you represent them into a network. If you use the method like the one I showed you before to integrate the multi-omics data, because we really work on extracting the correlation structure between those data, you can see that it's the Pearson correlation matrix is much more defined. And when you represent it into a network, it's very clear how those different omics features are correlated together. And so just to conclude on that, what we found is that when you do a multi-omics integration, you have new biological insights that you probably missed out from single analysis, because it's such a subtle signal. It can only be reinforced when you have some correlation happening between the different features. But we also find common information with the single omics analysis. So that really also helps to confirm some of the results. So I skipped the next example. Actually, it was an example in microbiome and how we did meta-transcriptomics, meta-proteomics, and 16S on a study in type 1 diabetes. But I will skip this. And just say, I want to thank people in my lab, but there's also a lot of people who have been working with me throughout the years, and especially to develop the mixomics tool. Thank you.
Thank you so much, Kim-Anh, for your wonderful sharing. I hope everyone else in the room will find your talk useful as much as I do. It's always, very difficult to understand the concept of integrating the multi-omics. So for the audience, if you are still here, if you have questions for Professor Kim-Anh, you can feel free to put the questions in the Q&A chat box. Otherwise, if you have afterthoughts, when you think about it throughout the days, you can always reach out to me if you want to get connected to whether to Kim-Anh or to Professor Mohammed. And let's see whether you can keep a few minutes, see whether there are questions flowing in. Well, maybe waiting for the first question to show up, or maybe I can ask Kim-Anh, like when I've tried to explore this, and then when you look through the literature, there's so many various like packages, right? Multi-groups, various groups to do almost similar analysis. Like just now, Professor Mohammed talked about the DADA tool versus the CHIMI. So what are the criteria you think will be important for us to use when making the selection, which one to pursue, especially when these are continuously being developed and being improved over time?
Yeah, I think keeping up with the methods that are developed is important. So some people tend to use the tool that has been around for a long time because they say, well, it's been developed for a long time, therefore, I should trust this. And I agree with that. You need to make sure that, the method has been there for quite a few years and it has been used by other people. Especially in R, when you use R, because a lot of people develop R packages, but then they're not very well. And you can see there's no example, like you don't know how to use it. There's no explanation about what you're supposed to do here. But I think it's really important too to monitor the literature and see whether there are studies that are similar to yours, and see what are the tools that have been used. Of course, there's a danger in that then people end up using always the same tool just because they think, well, if such and such used it and they publish in Nature, therefore it has to be a good tool. And sometimes they're not. So yeah, it's a bit of both, I think, is what seems to be the consensus in the past few years, as well as, is this tool well maintained? Are there people behind those tools? Are they talking to us? Like, is there a way we can contact them and have a feeling about this? Because there's many tools out there that are not maintained anymore. So if you have a bug or, if there's just issues, you won't be able to resolve it.
Yeah, that's a very good point. Thanks for your advice. Can I have I think this one Q&A. Let me see what's it. For anonymous. All right. So hi, Prof. If different techniques are used to analyze the omics data, then are these data comparable? For example, there's one paper on diabetes using
well, I can answer on that one because I've used it. You will have different data, and that's a problem. For example, my experience with Chung is that I would have many more OTUs than using the data too. It feels like you're missing out some information. You don't know why. I mean, you have to read about those methods, what is their advantage or not. It seems the consensus now is to use data too. But if you are in doubt, I would say use both. Do your analysis, see what your results are. And if you feel that there's, that one makes a lot of sense, the other one not so much, then that will actually decide for you what is the path you should take.
Okay, thank you so much for your answer. So, folks, keep your raw data. Your raw data will be useful to you for some time, right? Because you can keep mining.
Yeah. So what I want to say, and I think we've been discussing that with Ivy quite a bit. Is it's not in an analysis pipeline you have all these different steps: normalization, processing, normalization, analysis, and then you have your results. And those steps are not independent of each other. And so, in order to make sure that the whole pipeline is actually working, you have to get to the end and look at your results, and then you decide, oh yeah, that was good, or maybe it's a normalization that went wrong. And that's something that we don't tend to think. We think, well, once I do step one, I move on to step two, and everything's validated, and like you freeze your analysis and you go on, but that's actually not how it works.
Very linearized approach we often take. Yeah, thanks for the question. Any more questions? I think, well, Prof, if there are any questions, I will have your permission to channel that to you through emails. So, so audience, feel free, really. It's really important for us to understand before we venture into this very exciting part of research methodology. Okay, I think we are exactly at 9 over here in Malaysia. So I really want to thank Professor Kim-Anh Lee-Kao for joining us today from Melbourne, also Professor Mohammed. So, well, thank you so much for joining us, everyone. And I hope you will see you again next week for the next session or next episode of Breakfast at UMHealth. So I'd like to thank everyone again and take this opportunity to wish Selamat Hari Raya Haji for all, happy holidays and safe travel. Thank you, Professor Kim-Anh, for your time.
Bye.
Bye-bye.